When we get inside the forest, what we tend to see are big trees, beautiful giant ferns, grasses, and huge diversity of wild animals. However, hidden from our sight are very unique and vital treasures that drive and control the function of entire ecosystems. Macrofungi, mostly known by many as mushrooms, are unique living organisms driving the function of the ecosystem.
Fungi are a group of living organisms classified in their own kingdom fungi. Unlike plants, fungi do not make their own food. They are usually heterotrophic organisms. This means that they are organisms that get food from other organisms. There are usually two categories of fungi. We have the microfungi. Those are fungi we cannot see with our naked eyes. We need a microscope so that we can see them. And then we have the macrofungi. These are the fungi we can see with our naked eyes. And most of us know them as mushrooms.
Depending on the mode of nutrition, mushrooms in their natural ecosystems are either saprophytic, symbiotic, or parasitic. These three major categories of mushrooms enable to influence the structure, functioning, and dynamics of ecosystems processes and in the provision of ecosystem services.
Some of these fungi are saprophytic. That means they acquire food from dead and decaying organisms. When they are acquiring their food, they decompose the organism. And in the process of decomposing the organism, they release the nutrients into the soil. These nutrients are taken by the plants. And when they do this, they help maintain a stable plant community in our ecosystem. We have those which form mutualistic relationship with the plant species. In this relationship, both the plant and the fungi benefit. The fungi get the food from the plant, while the plant benefits by improved uptake of nutrients from the soil, especially the less mobile nutrients like phosphorus. They also help the plant by uptaking water from the soil. These fungi also help the plant by protecting it from or against the root-related diseases. When these fungi do so, they help to maintain a stable and healthy ecosystem or plant biodiversity. Other in their nature, they are parasitic. This means they acquire their food from a living organism. And when they, in the process of acquiring the food from the living organism, they usually cause diseases, and they are responsible for huge economic losses of our crops or even our trees. Therefore, it's very important to understand this diversity so that we can be able to manage our crops and our trees so that we can maintain a productive agriculture and a productive forestry sector.
Mushrooms are emerging as a viable crop in Kenya. They are highly nutritious sources of protein, vitamins, and minerals, and also cheap and easy to cultivate, which is compatible with small-scale farming systems.
So it is helping to improve the food security in our country.
Throughout history of mankind, mushrooms have been used for treatment of various diseases including cancer.
We have the medicinal mushrooms, which are very important in the medicine industry. We have mushrooms which produce dye. They are very important in dye industry or even in the clothing and textile industry. So mushrooms are very key and they are very important.
Despite their importance, mushrooms remain poorly known in Kenya and the world at large. Globally, we have over 5 million species of fungi. However, only about 250 species of fungi are currently documented and known. Over 4.75 million fungi therefore remain unknown. Out of these, only 72,000 mushroom species have been documented. In Kenya, 50,000 to 100,000 fungi species are proposed to occur. However, only less than 2,000 mushroom species have been documented thus far. This means huge numbers of mushroom species remain unknown, making their management, conservation and utilization difficult.
Biodiversity is the variety of the different form of life on Earth, and these include animals, plants, and even fungi, among others. This project, therefore, is going to put a lot of highlights on at least which are some of these species that we have in Kenya.
There is therefore urgency so that we can understand this diversity. We want to understand the diversity so that we can come with the strategies of their utilization. We want to know how to manage them, and we also want to know how to protect them or how to conserve them.
Unlike other living organisms, macrofungi are considered a strange group of organisms which are difficult to study due to their largely hidden nature, sporadic and random appearance during rainy season, and short-lived fruiting bodies.
Mushrooms are very unique species. They are unique in such a way that they have a very short lifespan. They have a lifespan of 0 to 20 days. They are affected by season. They mainly occur during the wet season or during the rainy season. Not all of them will appear at the same time. We'll have some which will be appearing at the beginning of the season, others at the middle, others at the end. So it makes work of documenting these mushrooms sometimes hectic. And the short surveys carried by scientists, they usually underestimate the diversity of these mushrooms in our ecosystem.
To document mushroom communities in Kenya, the National Museums of Kenya, in partnership with University of Nairobi, began a pilot project to test the effectiveness of citizen science and iNaturalist data collection tools in mushroom research and conservation. Through the generous grant from the National Geographical Society, the project engaged citizen scientists in documentation of macrofungi communities in Kenya.
We were exploring other methods which we can use so that we can understand the diversity of this mushroom in our ecosystem. And we found that we can engage the local community in form of citizen scientists. Citizen science is usually a program that engages the general public and involves them in correction of scientific data. In early 2019, we came in and we engaged some of the local conservationists so that they can help us document the mushroom in our ecosystem. We trained some of these local conservationists, some of students, some farmers. We trained them on how to correct the mushrooms, and we have engaged the citizen scientists in 2 ways. One of them, we have trained some local conservationists to be correcting for us mushroom in plots which have been already demarcated in a natural ecosystem, and also into the other land uses like plantation forest, into agriculture systems like tea plantation. The other method is that we have engaged citizen scientists all over Kenya, and these citizen scientists are engaged in such a way that they can use a mobile app called iNaturalist, and through this mobile app they can upload the observation they can see from their farms, from the areas where they are staying, and upload them.
We do conservation work in this Kereita forest, whereby we protect and we also monitor and do the forest policing for any checking on how the forest is behaving. So basically here, since we joined members of the National Museums of Kenya, we've been learning a lot on the mushrooms, conservation and protection. We've learned so many species of the mushrooms that we didn't know before.
This is the field guide book we use while collecting the mushrooms, and this one have helped me in identifying the mushrooms and also have sharpened my guiding skills.
We get into the forest and we have sample plots whereby every year during the rainy season, together with my colleagues, we are able to get there and collect mushroom species.
We go straight to the plot. We take the mushrooms which have already emerged. We take photos using this phone. Then from there, we describe how it is, the gills and the strap, and also the shape. Then from there, we send to the eye naturalist.
Our role has been educating other local communities here about those that are edible, and we have seen people going and looking for these edible mushrooms, and they're able to consume them.
Through this initiative, citizen scientists have discovered a wide range of mushroom species and generated enormous mushroom diversity data across Kenya. Over 1,000 mushroom species have been documented, some of which are new to science while others are the first records in Kenya.
Since then, we have seen a very great progress. We've had over 4,000 observations of mushroom, which we have been submitted to the National Museums of Kenya. And we can translate this to over 1,000 species of mushroom. So we can see the progress the citizen science program is producing to us scientists, because we are now able to understand the mushroom diversity in our ecosystems.
The National Museums of Kenya preserves dried and live mushroom cultures for future research and for use in mushroom and afforestation programs. Citizen scientists can contribute to this work of conserving and protecting mushrooms by collecting and submitting quality mushroom specimen and photographs to East African Herbarium, National Museums of Kenya. Citizen scientists can do this through simple collection methods.
Some of the things that we usually carry is a notebook. We require this notebook because we are going to record some information before we do the collection. We need a ruler because we have to measure the dimensions of these mushrooms. These are collection papers, and we call them greaseproof papers. So when we collect a sample, we have to label it. We have this equipment. We can use a shovel or we can use a bread knife because you have to uproot these mushrooms. So we have the cup. This is known as the cup. Then you have this part underside. These are the gills or the gill region. This is the reproductive part of the mushroom. Then you have the stipe. For plants, you'd call it a stem. We have this part, is known as the ring.
The first thing after you spot some mushrooms, the first thing you do is to take the coordinates of that area. The next thing, it's taking a good photograph. Make sure you are taking a photograph of the best mushroom or the mushroom that is in a good condition, not broken, not rotting, and make sure you capture its habitat. So the next thing, you make sure you uproot the mushroom from the base, the mycelia. Make sure you take all the details. Wow. And now we're going to take a photo of the underneath. Make sure you take the length of the stipe also, together with the tag name. It's describing the mushroom. Broadly convex shape. The pink. The surface. Oh, there. What about the margin? It's a little bit wavy. And there's a layer that is peeling off. So we go to the next step, the gills. They are the poles and they are numerous. What else? The color? Yellow. Are they attached to the stipe? Yes, they are attached to the stipe. You can also take the length of the stipe. In our case, our stipe is 3 centimeters. It's also thick and not hollow. It doesn't have a ling. It's growing on the ground. Pine plantation habitat. Something else, the number. How many are they? 1, 2, 3, 4, 5, 6. 6 mushrooms. Then you lap them. So there is a specific way we lap mushrooms to ensure that we don't damage them. So they are ready to be taken to the dryer to dry, and you are good to go.
Citizen scientists can also contribute to mushroom research and conservation by uploading quality mushroom photographs in iNaturalist mobile or web application. iNaturalist is a citizen science project and online social network of naturalists, citizen scientists, ecologists, and biologists, and is built on the concept of mapping and sharing observation of biodiversity across the globe. If you are new to iNaturalist, iNaturalist may be accessed through its website or from a mobile app. You can do this by downloading iNaturalist app into your smartphone or even computer from Google Play Store or Apple Store. For example, if I have Android, I go to Play Store and then I select or choose iNaturalist by typing iNaturalist and then select iNaturalist, and then you upload. Once uploaded, you can now open the application so as to be able to take your photos. But make sure that before you take any photo, that your GPS location is on by clicking the location. And this is to capture the geographical location of your observations. So once you have uploaded your iNaturalist, that is from Google app or Apple app or store, then it is possible now to come and then you can be able to click iNaturalist icon. Then it will take you, like for example, mine, because I'm already registered, I have my observation, but I want now to make a new observation. So I click at the add here plus, so that now I can be able to take a photo. So I click at take a photo, and then it will take me to my photo. So this is my observation from the field, which I've already collected, and then I can take a nice photo of the same. And then you can say, click OK, that is the tick, yes. And then it is going to prepare the photo. It has already prepared the photo. And once you find that the photo is okay, you can add notes. For example, I took this from Kereita Forest. So I can add Kereita there forest, for example. Or you can also add as much details as possible and agree. Take another photo from the same area or from the same specimen. Like I want now to see the gills. So again now, I can take now the gills of the same, and then I say okay. So now I can take another photo of the same, but now to be able to show the gills. Again I say okay. So I have 2 photos. You can see. You can also suggest the name of your observations. So what you do, just below the images or your image, you can see where it is written, what did you see? So you can view suggestion, and because as we are involved or concerned with mushrooms, so we can suggest the name fungi. So you write the name fungi. If your mobile has data or data is on, then it will be able to give you the various suggestions. So in our case here, we can select fungi including lichens. Otherwise, if you do not have data, you can just write the word fungi and then accept that alternative by clicking this tick here, and the fungi including lichens there, and then down here the green tick you accept by clicking the tick there, and then obviously it has now put the suggestion. And you can even add, because our project is called Mushrooms Project or Mushrooms of Kenya Project, so I can highlight so that I will add that specimen, that photo or those two photos into our Mushroom of Kenya project, like I have done. So it has added itself, so it will appear in the Mushrooms project. Even though now it is unknown, we'll have so many observers who will help in identifying this species for ourselves. Then we can use that information to enrich our database.
At the East African Herbarium, submitted mushroom specimens are properly identified, dried, assigned a unique collection number for reference, and stored in cupboards for future research and use.
We start the process of identifying the species, the specimen that we have brought, and also preparing them so that we can preserve them. We usually get a sample from our basket, and then we actually unfold it from the way we had wrapped it. Then from the sample, I'll pick one of the samples because for the process of identification, I require to do what we call a spore print. Because a spore print gives us different colors for different species. So what I normally do is that I get a sample like this and then I remove the stipe from the cup. And so what I do is that I get a white paper. So I place the cup with the underside of the cup facing downwards. Then after that, I come with a bottle. So I place it on top of the cup. And I'm going to leave this overnight. And I expect that after a night, I'm able to see a print like this. Then after that, I go to the next step because we want to dry these specimens. You realize that 90% of mushroom is water. So we have to cut it, and especially for these huge mushroom specimens, we dissect them. We place them on grease-proof papers. At the same time, we would want to carry out some more work with these mushrooms. So we usually preserve like a section of it, or even one sample, because some are edible, and we would want them to actually culture them in artificial media. So I set a representative of the specimen, I preserve it so that I can do further work of culturing it. And at the same time, I can use this sample to get a section of it for the molecular analysis. So after that, okay, I wrap the specimens which I have sectioned so that I can take them down to the dryer. And I expect that after 3 days that they are going to be dry. These are some of the specimens that have been dried using this method, as we can say, for many years. So this is the continuation of identification, and as I've said, we do microscopic work, and this is where we mount very thin sections on a slide. Then we cover with a cover slip, and then we want to look at certain features, because for different species they have different features. That one usually helps in the process of identification. And so as I look at different microscopic morphologies, then I also enter that data, and that data helps me in the identification of my species.
The long-term goal of this project is to ensure sustainable management and conservation of mushroom resources in Kenya. We are currently losing a huge number of mushroom species when mushroom natural habitat are degraded through unsustainable human activities, or when natural ecosystems are converted to other land uses such as agriculture, plantation forests, industrial parks, mining farms, and urban centers, or sheer deforestation.
When we convert an indigenous forest to plantation forest, we lose around 90% of our mushroom. And when we convert natural forest to agriculture, we lose about 99% of this mushroom. Therefore, it is very important we have method and strategies of protecting this mushroom from loss or from being threatened in our ecosystem. We have two methods of protecting our mushrooms. One of them is in situ conservation method. This method, they are usually conserved in their natural condition.
Things like, for example, our national parks, our game reserve, our conservancies, whereby we have number of game reserves which have been set up by the government, at least to be able to conserve biodiversity, including mushrooms. We also have other areas, for example, like conservancies, which again can help quite a lot to conserve our mushrooms. We have even a roadside, we have forest, we have grasslands, and especially protected forest, like where we are doing our work at Kereita Forest, which is part of the southern Aberdares Forest. In addition, we need to see how we can stop removal of substrates. When we end up removing some of these, maybe our local communities are removing them as firewood, then obviously you find that we continue to threaten our mushrooms.
And there is the second method, the ex situ method of conservation. This is where we usually take the mushroom from their natural state, and we bring them now to the lab where they are conserved. One of the method of conserving mushroom for future research is to do the tissue culture of the mushroom. We want to culture it in a media. Here at the museum, we usually use two types of media. The PDA, which is the potato, dextroso alga, or the MEA, which is the malt extract agar. So when you have made and prepared your agar, we usually put it in a petri dish like this one. We make sure we get a tissue which is inside the mushroom, which is very clean and which is not contaminated. So the first step we do is we sterilize the cup with the 70% alcohol. After sterilizing the cup, we come with our blend, and in this blend you have to sterilize it both in 70% alcohol and also in the ramp. You have to heat your blend until it is red hot. You come and cut your specimen. So we try to get the tissue from deep within the mushroom. We press it now in our agar. Then we have to close our media to make it airtight so that no contamination enters. After that, we store this media in an incubator at a 25 degrees centigrade. This is the incubator. We first set the temperature. You put it at 25 degrees. Then you come and put your tissue catcher. You keep it there and you keep monitoring so that you ensure it's growing. When we are satisfied that the growth is sufficient, it can now be used to make the mushroom seed, which is called the spawn. And that spawn now can be used for mushroom cultivation. It can be used for making the inoculum for restoration programs. And at the same time now, these ones we can store them in liquid nitrogen. We can take them to the gene bank where they are stored for future research. And now from there we have our catcher, and we can now be able to use our mushrooms, which we have gotten from the field, for anything that we desire to use those mushrooms for.
Come, let us join hands in making our mushroom diversity known to enable sustainable management of mushrooms for healthy and productive ecosystems.